it's over to you.
Okay, thank you very much, Thomas. Dzień dobry Państwu. Witam na naszym szkoleniu z oprogramowania VarSome. Krótki wstęp, następnie przekażę głos Tomasowi, który pokaże Państwu możliwości tego oprogramowania. Jako Perlan podpisaliśmy kontrakt dystrybucyjny z firmą VarSome 2 miesiące z firmą VarSome na terenie Polski. Także jeżeli będą Państwo zainteresowani, to zachęcamy do kontaktu z nami albo bezpośrednio z Tomasem. Jako Perlan doskonale Państwo wiedzą, że oferujemy również różnego rodzaju rozwiązania do biologii molekularnej, głównie do NGS od firmy Agilent. Także paneli na życzenie, czyli program Sure Design. Mieliśmy możliwość wyboru technologii do tworzenia bibliotek Sure Select albo też Haloplex. Mieliśmy również gotowe zestawy do sekwencjonowania z marki Multiplicom, która to została kupiona przez firmę Agilent już jakiś czas temu. Jak również posiadaliśmy dzięki Agilentowi kolejne narzędzia do kontroli jakości bibliotek oraz do analizy bioinformatycznej, czyli tutaj program SureCall oraz zestaw oprogramowania ALISA. Było to firmy Agilent, dlatego poszukiwaliśmy programu, który byłby otwarty na różnego rodzaju zestawy odczynnikowe, z których możemy uzyskać biblioteki NGS. W związku z tym rozpoczęliśmy współpracę właśnie z firmą VarSome, która jest otwarta na biblioteki uzyskiwane za pomocą różnego rodzaju odczynników i umożliwia to oprogramowanie analizę wyników z poziomu FASTQ oraz z poziomu VCF. O szczegółach za chwilkę opowie Tomasz. Również w tym miesiącu podpisaliśmy umowę dystrybucyjną z firmą Swift Biosciences, która udostępnia amplikonowe zestawy do tworzenia bibliotek. I tutaj firma Swift oraz VarSome już współpracują między sobą na zasadzie takiej, że Swift dostarcza odczynniki do tworzenia bibliotek, a VarSome umożliwia analizę wyników z tychże właśnie zestawów. Także jako Perlan jesteśmy obecnie widzą na sajcie. Krótko o firmie Swift Biosciences, dlaczego byliśmy nią zainteresowani. Głównie dlatego, że oferuje bardzo szybki protokół. Jak Państwo widzą na schemacie po prawej stronie, przygotowanie biblioteki do NGS trwa 2 godziny. Jest to jedna reakcja PCR, czyszczenie oraz jeszcze i Swift oferuje zarówno zestawy katalogowe, jak i tworzenie swoich własnych paneli na życzenie do rozmiaru około 100 000 par zasad. Także jeżeli są Państwo zainteresowani większymi panelami, to wracamy w tym momencie do technologii Agilent, panele na zasadzie Sure Select, które dadzą nam dobre wyniki. Zaletą Swift jest również to, że cała reakcja zachodzi w jednej probówce. W odróżnieniu do innych zestawów amplikonowych nie ma potrzeby dzielenia jednego DNA od jednego pacjenta na kilka próbek, tylko wszystko idzie w jednej probóweczce, tak jak wspomniałem, jedna reakcja PCR. I specjalnością Swift jest to, iż wszelkie trudne matryce, czy to jeżeli chodzi o małe stężenia, czy to jeżeli chodzi o jakość tego materiału, nadają się do zastosowania do tych paneli amplikonowych. Możemy wykorzystać i dobrać panele do tradycyjnego, powiedzmy, DNA-seku, tak, gdzie mamy listę genów i chcemy je sekwencjonować. Może to być materiał izolowany z parafiny, może to być wolno krążący nowotworowy DNA, może to być DNA z materiału kopalnego. Są też dedykowane zestawy do badań transkryptomicznych, czyli do RNA-seeku i również do badania metylacji DNA z bardzo małych ilości wejściowego DNA, również z wolno krążącego nowotworowego DNA. Jest też osobny zestaw do badania mikrobiomu, czyli do 16S, gdzie są pokryte wszystkie regiony od V1 do V9, dzięki czemu dostaniemy pełną informację o tym, co się znajduje w naszej próbce. Tak jak wspomniałem, w przypadku tradycyjnego DNA-seku możliwa jest analiza wyników w oprogramowaniu VarSome. Także według mnie 3 najważniejsze cechy programu VarSome to jest to, iż możemy zakupić licencję już na analizę pojedynczych prób. Nie ma potrzeby inwestowania od razu w jakąś dużą licencję, tak jak ma to miejsce na przykład w przypadku programu ALISA, więc nawet pojedynczą próbkę możemy przeanalizować za pomocą VarSome. Dodatkowo, jeżeli mamy wyniki z platformy Illumina, to możemy pracować na wynikach FastQ albo VCF. Jeżeli mamy wyniki z platformy Thermo, możemy pracować tylko na wynikach VCF. I jako że mamy w dystrybucji te wszystkie wspomniane 3 firmy, to możemy powiązać ze sobą licencję na przykład na program VarSome razem z zakupem odczynników, czy to Agilent, czy to Swift Biosciences. Oczywiście VarSome może być użyty również do analizy innych wyników z innych firm, tak, na przykład Deviser, więc jeżeli Państwo są tym zainteresowani, to również można zakupić VarSome i analizować wyniki z tamtych firm. Także dziękuję bardzo Państwu za uwagę. Oddaję głos teraz Tomasowi. Thomas, it's up to you right now.
All right, Wojciech, thank you very much for a very nice introduction to Perlan and to your services. So my name is Thomas. I work for VarSome. I'm the Director of Business Development. And I'd like to tell you a little bit more about our company and about the tools we are developing. Let me figure out how can I share my screen first. Share. All right, Wojtek, could you confirm you see my screen?
Yes, it's visible. It's fine.
All right, great. All right, so thank you very much again, Wojtek. And so we are a bioinformatics company located in Switzerland, and we develop tools for processing and interpretation of NGS data, specifically for clinical purposes. We have created basically, we have several platforms. The first one, it's called VarSome, and this is the one which you can see on your screen at the moment. So VarSome, and I guess, you know, at least some of you have seen it before, VarSome, it's a free platform. It's a search engine which aggregates and cross-references publicly available data. Yeah? And so at the moment, we have integrated over 35 data resources, such as ClinVar, DVSNP, Genome ID, Genome AD, Ensemble, RevSeq, somatic data resources, and so on. Simply said, over 35 data resources linked together into a single, cohesive knowledge base, which we call VarSome. Secondly, VarSome allows you to perform queries against this knowledge base consisting of 35 data resources. And so on VarSome, you can query, you know, for RSIDs, you can query AGVS terms, you can look up variants based on chromosomal location. You can also look up genes and phenotypes. So in fact, it functions a little bit like a Google. That's the reason why you can think of it as a Google of genomics. Yeah? And so, for example, here you can see results for a particular variant in a BRAF gene here. Yeah. And so I'd like to guide you very quickly through the result page here. And so first, of course, you can switch between two reference genomes, AG19 and 38. And then, of course, here you can see the common information for the variants, such as chromosomal position, reference sequence, and so on. And then here, right beneath, you can see a really important component of VarSome, and that's the automated interpretation of pathogenicity based on ACMG guidelines. And so in this case, for example, in the case of this variant here, you can see that 5 criteria fired based on the aggregated evidence coming from all these 35 data resources. In other words, our implementation of ACMG guidelines, it's very robust. It's very solid. Why? Well, because it's based on top of all these, you know, again, 35 data resources linked and cross-referenced together. Secondly, it's also a very transparent interpretation of ACMG guidelines, because here, for each criterion, it tells you in a human readable format why given criterion fired or didn't fire. You can also click on the blue question mark here, and it tells you, the same thing. Of course, not all the criteria can be fully automated, right? Certain criteria require a patient-specific information or information about allele segregation. And so in those cases, it's you who has to make, the judgment call here. So you can always modify the criteria based on your private knowledge for that particular patient, for example. And so as you check and uncheck, certain criteria, to verdict updates dynamically. You can even select the transcript for annotation, for annotations, which obviously can affect the final verdict as well. Going farther down the page here, for example, you can see the gene landscape, which summarizes all the variants within the given gene. So here you can see there's quite a few pathogenic variants, for example, so there is probably a mutational hotspot. And so for each variant here, you can see the supporting evidence coming from all these data resources, such as ICGC, ClinVar, UniPro, DBSNP, and so on. Here is another really important aspect of VarSome, of the free platform, and that's our global community. VarSome, it's a free platform, right? And so we have over 175,000 users globally using it every day. And so we have a growing number of contributions from our global community. Actually, we encourage, VarSome users to provide something back in exchange for providing the service for free. And so there's a growing number of contributions, such as publication links. Our users link functional publications, functional studies, classify variants, start discussions, or even start new collaborations. And then in turn, these contributions from our global community we leverage for certain ACMG criteria, such as is the case of criterion, for example, PP5. So here you can see that the PP5 criterion considers ClinVar entries as well as contributions from our global community. So VarSome, it's not only about the public data resources, it's also about gathering, contributions from our global community, which constitutes an absolutely unique layer of data, which is not available elsewhere. Going farther down the page here, you can see list of publications, of course, coming from ClinVar, Civic, or PMKB. So PMKB, it's especially important source of data specifically for somatic samples or for somatic cases. PMKB stands for the Precision Medicine Knowledge Base maintained by the Weill Cornell Institute in New York, which also includes links for clinical trials as well as drug-related information, as you will see a bit later. Again, here you can see structural variant browser with all the structural variants coming from Decipher, DGV, ExactCNV, or ClinVar, all the transcripts from RefSeq or from Ensemble, ClinVar entries. So this is a particularly well-studied variant in a BRAF gene. So there is quite a few entries coming from ClinVar, UniProt variants, allelic frequencies from the latest version of GNOME AD project, the version 2.1, broken down by ethnicities and gender. PMKB, right here you can see, now you are going to see a number of somatic data resources such as PMKB, which also displays drugs, for example, and links for clinical trials. ICGC, another important data resource for somatic variation. Cosmic, Civic, genomic data commands. And at the end of the page, you can see a number of in silico prediction scores as well as conservation scores. So on a single page, as you can see, on a single page, you can access, the results from all these 35 data resources. So VarSome, it's a very, very convenient way how to look up a comprehensive information for a single variant. It's a freely available platform, so I encourage you, to check it out and to see what it does for you. So this is the free platform. However, the objective of our call today is to present you our platform called VarSome Clinical. And so VarSome Clinical, it's a kind of an extension built on top of the free platform. And so VarSome Clinical here, it's a complete solution from processing and interpretation of NGS data, starting from FastQ or VCF. And so here, I'd like to tell you a little bit more about it. So as I've just mentioned, we are a bioinformatics company located in Switzerland, and that's also the place where we have our physical servers located under our full control in Switzerland. And so that's the place where our clients upload the raw sequencing data, FastQ files, or alternatively VCF files. Once you upload the data, you can run the pipeline. We offer a wide range of pipelines for germline samples, for somatic samples. We have a pipeline for copy number variation, structural variation. We can also have a pipeline for de novo variants in trios, for carrier risk in couples, somatic germline pairs. So simply said, a wide range of pipelines covering pretty much all the clinical use cases. VarSome Clinical comes with a website interface, which means you don't need to install anything. Everything runs on our server in Switzerland, and you access the platform over the internet using your website browser. And at the end of the process, you have the option to generate a clinical report. Speaking of compliance, VarSome Clinical is certified for medical use. It's certified as an in vitro diagnostic medical device according to the requirements for CE-IVD marking. So that's quite, of course, an important aspect for clinical use. And also our company is certified with 2 ISO certificates, one for data quality or quality management, and the other one for the 27001 for that data security, I believe. And on top of it, we are based in Switzerland, which is a country, as you know, which is a country very, especially sensitive towards privacy and security. And so there's a number of other local requirements in Switzerland, which we have to comply. In other words, your data are safe with us. Speaking of sensitivity and precision, or speaking of performance of our pipeline, as you can see, it's quite high, over 99.8% for SNPs and indels. And these are not made up numbers. These are the results from the precision contest. So precision contest, it's basically a competition, a bioinformatics competition organized by FDA, the Food and Drug Administration Office in the US. And that's where we regularly participate. And these numbers are the results from the latest run of the precision contest. Okay, so this is just a very brief introduction to our company and to our 2 products, the free platform and called VarSome, and then the other one, VarSome Clinical, which is a complete solution for processing of NGS data. All right, now I think I'd like to proceed, and I'd like to show you how the real VarSome Clinical looks like and how it works. However, before I start, I've just realized that I forgot to mention that you can ask questions. Yeah, so here on the navigation bar, you can see a section for questions and answers. So whenever you have a question, please type it in, and later we will answer and discuss all your possible questions. And so here you can see the real VarSome Clinical. So as I've just mentioned, the first step consists of uploading the data. So first you have to upload data to our server in Switzerland. So you can select your data directly from your computer, right? And send it over the internet to our server in Switzerland. There is also an API. Basically, for every step in the process, there is an API endpoint, which is a bit technical thing. In other words, API allows you to automate a certain aspect of the pipeline. So just remember, there is also API available for every step of the process, including for uploading the data. Once you upload the data, you can run the analysis. So here you can see two options, starting from FastQ or VCF. Also, with VarSome Clinical, you can process any kind of NGS data, be it a gene panel, customized gene panel, or off-shell assay, or exome or genome. It doesn't really matter. The only requirement for FastQ files is that you shall sequence on Illumina machine. Yeah, so FastQ files shall be produced, on an Illumina machine. When it comes to a VCF file, we can take any VCF file as long as the VCF file conforms to the standards for VCF files. So that can be PathBio, Thermo Fisher, you name it. And so here we are back setting up the analysis here. So first we have to specify the pipeline, whether it's a single sample, family trio, couple for carrier risk screening, generic multiple sample for extended family, or for cohort studies. So you have to specify whether it's a germline sample or somatic sample here. Here you have to specify the assay, and so that would be, for example, Swift Biosciences, right? As Wojciech mentioned during his presentation, basically we have become partners with Swift Biosciences. Swift Biosciences is an assay manufacturer. They produce amplicon-based assays for a number of use cases here. And what we do is that we bundle our products together. The Swift assay bundled with VarSome Clinical for data interpretation. And so this way we can deliver a really complete solution, right? Starting with chemicals and ending up with clinical report. And this bundled solution is later distributed and supported by Perlan, by our joint distributor for Poland. And so this is what you have to specify here, the kind of assay, but also you can process any kind of NGS data, right? Be it the Agilent assay or Twist exome or complete genome or whatever. So it's really flexible here. Here you have to specify the ethnicity of your sample or of the patients. You can also provide optional details for sequencer. You can select the reference genome, AG19 and 38. Here you can specify whether you want to call all the variants or only the variants that pass certain quality filters. Here is another important option where you can control whether you want to perform a full analysis or gene list restricted analysis. And so here you can set up a gene list. So you can either set it up manually just by copying and pasting the list of genes, or you can set it up automatically using HPO terms, the human phenotype ontology. And so here you type in the phenotype of your interest, such as osteopenia, and so here you can see that according to HPO, this phenotype is associated with all these genes. And so this is the way how you can set up very quickly a specific gene list for a specific set of phenotypes, and subsequently you can use it as a filter for your analysis. The use case for this option here is to be able to avoid incidental findings. Incidental findings, especially when it comes to a larger data sets, such as exomes or genomes. And then on the left side here, you have to specify the sample details, such as the source data, the raw data for the pipeline. You can also provide a sample ID, sample description. Here you can type in the phenotypes and diseases, again, according to HPO for that particular patient. And later later in the process, you can filter your variants based on these terms here. Once you set up all these details, you hit the start button and the analysis starts. Speaking of performance, the process is very fast. Just to give you an idea, how fast it is. So an exome, a whole exome takes about half an hour. A whole genome takes about 4 hours, and the gene panels, it's a matter of 5 minutes, between 5 and 10 minutes, depending on the size of the gene panel. So it's pretty fast. And once the analysis is finished, you of course can access the results here. And so I'm going to show you, first the whole genome sample. I'm going to show you a number of samples during my presentation, but I'd like to start with a whole genome sample for now. And so here you can see, let me resize the screen a bit here. Okay, so this is a whole genome sample with almost 5 million variants. Quite typical genome, I guess. As you can see, the variants are sorted by pathogenicity according to the ACMG criteria. Yeah? So this is the ACMG verdict in the column here. Apart from that column here, you can see a number of other columns, of course, such as variant name, variant type. Here you can see a column for custom classification. And so this is really important feature of VarSome Clinical. So according to the ACMG, this is a pathogenic variant, right? But of course, if you don't agree with the verdict for whatever reason, you can reclassify the variant internally. You can reclassify it as a benign or likely benign variant, for example, internally, privately, only within your account. Yeah? And then this reclassification stays with the variant forever, basically, unless you change it or remove it. And so that means that whenever you upload a new sample with the same variant, you will see the same classification mark here, including the information who did it. Okay. And you can even set up a fully custom classifications. You can mark a variant, as a variant important from a pharmacogenomics perspective, or a variant that needs to be discussed with the lab director. Yeah? You can set up any classification you like, and again, you can apply these custom classifications to your variants here, and again, these classifications stay with the variant, and whenever you upload a new sample, you will see the same classification mark. You can even, for example, here modify the list of ACMG criteria applied to a given variant. So here, by default, you can see that 4 criteria fired. But of course, certain criteria require, again, a patient-specific information, for example, or information about prevalencies and incidences, such as the case of VS1. And so basically, you can override here and the default ACMG verdict here, and then you can save it as a manual classification. And that's what you can see again here in this column for custom classification. So in the case of this variant, you can see that, 5 criteria has been modified manually. So this is, something that what allows you to build over time your private database of classifications. I guess you might have a manually curated list of variants in a spreadsheet, I guess. And that's a common situation. And so what we can do in those situations is that we can take your list of manually curated variants and upload it privately to VarSome Clinical, for the purposes of variant annotations. Going farther down the variant table here, you can see the list of ACMG criteria that fired for each particular variant, AGVS term, exon number, gene, mode of inheritance, function, whether it's coding, missense, splicing, non-coding variant, frameshift variant. We annotate here in terms of function, we annotate against all the transcripts available. When it comes to ACMG, we annotate against to the canonical transcript here. However, if you prefer, you know, to annotate against your preferred, your custom transcript, that's also an option. You can send us the list of genes with the list of transcripts, and we upload it again, you know, to VarSome Clinical. And zygosity, whether it's heterozygous, homozygous variant, frequency, this is the allelic frequency according to the ethnicity specified in the step before. Allelic balance, this is which is the percentage of reads supporting the given variant, and coverage, the number of reads covering the given variant. The coverage, it's a link, so you can click on it, and it takes you, you know, to the JBrowse view. This is a JBrowse where you can see the graphical representation of reads. Yes, so you can zoom out and zoom in. So this is the first place where you can go and check whether a particular exon has been fully covered or not. VarSome Clinical also comes with a number of QC reports, quality control reports. So for example, here in this drop-down menu, you can access, for example, you know, the basic QC report, which summarizes the details of the sample, the details of the software used for processing of the data, details of databases used for annotation and interpretation, summarization of the alignment step of the pipeline, summarization according to the coverage, according to the pathogenicity, the number of pathogenic and likely pathogenic variants, summarization according to each individual ACMG criterion, and at the end, summarization in terms of function, the number of missense, nonsense variants, and so on. So this is the basic QC report, which is also available here in a PDF version. However, there is a more comprehensive report available, which is called the coding. You can also download the BAM file or the indexed BAM file, for example, locally. And however, here is the most comprehensive report called the coding coverage report. And so here you have to first specify the list of genes for which you want to generate the coding coverage report, and later you get an Excel sheet, an Excel table with a very detailed coding coverage report broken down by individual genes, chromosomal position, and exon number, okay, and sequencing depth, of course. Okay. So this is the place where you can identify the regions with a low coverage or regions that have been skipped altogether for whatever reason. So this is the place where you want to go once you need to troubleshoot issues with sequencing, for example. Okay, and then around the variant table here, you can see a number of tabs. So for example, here you can see a tab with allelic frequencies, with clinical information, transcripts, all the transcripts coming from RefSeq, Ensemble, and so on, the list of genes associated with given variant, HPO terms again, cross-reference samples, I'll get back to it in a while, nearby variants, audit trail, so this is a bit, you know, a compliance related feature. Basically, VarSome Clinical logs every action performed by any user for auditing purposes. And then here beneath the variant table, here you can see the ACMG grid. Similarly to the free platform, right? Again, it tells you in a great detail why given criterion fired or didn't fire, and you can always, you know, again modify the verdict by triggering additional ACMG criteria according to your private information, private knowledge for that particular patient. A region browser again, which summarizes, you know, all the variants within the gene, ClinVar entries, all the ClinVar entries, cosmic entries, caviar entries, allelic frequencies from GNOME-AD, ICGC data, genomic data commands, in silico predictions, conservation scores, list of publications again, as well as contributions from our global community. in certain cases, of course, not for all the variants, but in case there is a contribution on the free platform for a particular variant, you will also see that information in this section here. So yeah, so this is the dashboard area where you can access, you know, the results, the primary results of the analysis. Do we have questions? All right, I see. Let me check questions. What is the sensitivity for detecting copy number variation? All right, I'll get back to this once I talk about the pipeline for CNVs. So I'll get back to it. Now we have 5 million variants, right? This is a genome, whole genome. And so we obviously need filters. We obviously need to prioritize somehow the variants. And so here on the left side, you can access filters, you know, the first kind. We have several kinds of filters. Here you can access the first kind. So let's create a filter. So with these filters, you can filter your variants based on a number of criteria, such as ACMG rules, or allele frequency, or predicted score, or ClinVar classification. You can filter your variants based on chromosomal position, based on pathogenicity, based on zygosity, based on function, whether it's 5UTR coding, splicing variant. Again, you can apply a gene list as a filter here, call status, and a couple of other options here. And so just to give you a quick example of, you know, how it works. So let's say we want to filter out only rare variants with a frequency less than 1%. So we are now setting up a filter set. Yeah. A filter set may consist of several filters, such as filter for frequency, and the other one can be a filter for pathogenicity, for pathogenic and likely pathogenic variants. So now we have 2 filters within the filter set. We give it a name, and once you set up a filter set, you can use it all over again for all your future samples. So basically, you can set up a multiple filter sets for each kind of sample, for example. Save, and now again, just, you know, please notice that, you know, everything happens really fast, right? So, so using the frequency filter, we have eliminated over 4.5 million variants based on the frequency, and based on the pathogenicity filter, we have eliminated 339,000 variants, and we are left only with 4 variants down from the original 5 million, right? And as you can see, everything happens really fast. That's why, that's because we have spent lots of time and resources for, you know, on performance aspects of the platform. So you can always, you know, deactivate and reactivate certain criteria in the filter set, or you can even go one step further and you can modify the whole filter set. And so, for example, you can also include a third criterion for ACMG, for PP5 rule. So for example, now we have 3 filters within the filter set. Again, we apply it to the sample. And as you can see, based on the ACMG filter for PP5, 2 variants were eliminated, and we are left with only 2 variants in the variant table. You know, so as you can see, you can easily modify, you know, these filters, and you get the results dynamically. Yeah, so that's the reason why we call these filters dynamic filters. Yeah, so dynamic filters are really easy to use, easy to modify, and you get the results instantly. So these are the dynamic filters. However, there is another kind of filters available called algorithmic filters. And so algorithmic filters are even more powerful than dynamic filters. You can think of it as a little programs. Yeah, algorithmic filters are basically little programs we build for each customer, depending on the customer's needs, yeah, and depending on the specific workflow for each particular customer. Yeah, and so these algorithmic filters offer a great deal of flexibility. Yeah, and so here you can see, for example, in the list of the most common algorithmic filters. Yeah, and so just to give you an idea of what's possible with algorithmic filters, for example, with algorithmic filters, for example, you can call de novo variants for trios, or you can do carrier risk for couples, or you can do compound heterozygous variants, or you can perform a segregation kind of analysis for dominant or for recessive variants. Yeah, and so these filters can be fully customized according to your specific workflow, and it's a part of the service, it's a part of the platform. And so what it does actually, in fact, is that algorithmic filter creates a snapshot of the original analysis, for example. Yeah. And so, for example, here you can see an exon trio with 290,000 variants. This is the parent analysis. And with him, you can see a number of sub-analyses. Yeah. And so each sub-analyze corresponds to the results of an algorithmic filter. Yeah. So algorithmic filter basically creates a snapshot of the original analysis. And so, for example, here you can see the results of an algorithmic filter for de novo variants in exon trio. Again, you can see the results on the dashboard area here. It's almost the same dashboard area as for single samples. However, there are slight differences. For example, the zygosity column displays the zygosity for each individual sample in the trio, the child, mother, and father, and the same for allelic balance and coverage. In the JBrowse, you would also see the alignment of reads for all 3 samples within the trio. So these are the results of an algorithmic filter for de novo variants, and then of course you can lay over the dynamic filters for allelic frequency, ACMG criteria, function, zygosity, and so on. So you can really easily combine these fully customizable algorithmic filters with these dynamic filters. Yeah, so filtering, it's really important. We spent lots of resources on building these filtering capabilities in VarSome Clinical. There is also a pipeline for copy number variation here. So again, copy number variation basically functions as an algorithmic filter, basically. So as you know, for example, copy number variation, calling CNVs on exome data or on gene panels, it's a challenge. There is no perfect solution for it, and so we use a number of tools. For example, we use exome depth for CNVs, and so for this tool you need to upload at least 5, ideally 10 samples sequenced during the same sequencing run. And then here you can see the results again. Here, a copy number variation is a sub-analysis. The amount of data, annotation data for CNVs, it's quite limited compared to SNPs and indels. However, that's what it is. And so here, for example, you can see, let's choose this one. So this particular CNV, this particular duplication lies between 2 red lines. And so here in this browser, you can see how well your CNV overlaps with CNVs coming from public data resources. The fact is that the CNVs very rarely overlap perfectly, right? So yeah, the amount of annotation data, for copy number variation, it's still quite limited. Now, I've just realized I have forgotten to mention a really important aspect of VarSome Clinical, and that's the sample cross-referencing here. And so what we do, as long as you are our client, what we do is that we link all your samples together on variant level. Yeah. And so for example, here you can see this particular variant has been found so far in a heterozygous form in 2 samples, in a sample called exome 7 and in this analysis. Yeah. So this is a demo account, right, with a limited number of samples. Yeah. So, however, once you start using the platform, you will see the list of cross-referenced, samples to grow. And so this is something what allows you to go back and forth between your past cases and current cases, compare the patient phenotypes, and ultimately increase the diagnostic yield for your patients. Yeah. So we do it on 2 levels. We cross-reference your samples on 2 levels, basically. We do the sample cross-referencing across your own samples. In those cases, we can always display, the details, the sample name, sample link, list of phenotypes, and so on, because those are your own samples. However, we also cross-reference the samples on the global level across all the samples coming from all our clients, and that's what you can see here. However, in those cases, we can display basically just the number of occurrences of a given variant among all the samples on the platform. So we cannot display, of course, any sensitive information, but still, we believe it still gives you additional clue for interpretation of pathogenicity for that given variant. What you can actually also do, so everything what you do here, it's private, right? The custom classification, sample cross-referencing, everything is private. It stays only within your account. However, you can optionally share it with your partner institution if there is a written agreement between these 2 institutions. Yeah, so VarSome Clinical also allows cross-institutional collaboration in order to increase the diagnostic yield. Okay, so now I think the last missing part is the reporting part here. So let me activate the filters again. So once you narrow down the list of variants using dynamic and algorithmic filters, you have to decide which variant you are going to report, right? And this is a manual process. You have to decide what are you going to report. And so once you decide to report the variant, you select it for export here. You can even set up comments. This little icon means there is a comment on the variant. And again, this comment stays with the variant, and later it also appears in the clinical report. And so once you select the variant for reporting, you can proceed and you can generate the clinical report. So the clinical report can be fully customized. You can replace the logo, you can update, the fonts, colors, and sizes. You can even translate it into any language. So basically, you just need to set up a template according to your branding policy, and then you can use it again for all your future samples. The reporting part of VarSome Clinical functions as a drag and drop editor. Yeah? And so here on the left, you can see the list of widgets available for each particular kind of pipeline. And so, for example, here you can drag in, there is already a widget for analysis information, right? It's already here. And so you can, for example, also include the content area for your comment, and then you can include the information about the variant, information about the gene, the list of drugs, and list of references. Yeah. And here we have it. Here's the content area where you can provide your custom comment for the report. Here's the information about the variant, the transcript and AGVS term, classification of it, the comment which comes with the variant, information coming from clinical databases, information about the gene, the list of phenotypes and diseases, mode of inheritance, a list of drugs, if there are any, as well as, you know, the list of clinical trials and the list of references here. So everything is fully editable. If you don't like this publication, or if you don't like these people, you can remove the publication from the list, and you can add your own publication for your own glory, for example. Yeah, so as you can see, it's a fully editable reporting feature here, and once you are done with it, you can download it as a PDF file, or as an Excel file, or as a doc file. So here, for example, you can see a couple of examples of the final report. Again, this is the default template without any styling. And so this is actually a specific example for a Swift Biosciences 56G assay for solid tumors, if I remember well. And so here you can see the list of variants, you know, in this 56G sample. Again, with the information about the genes, information coming from clinical genomic databases, list of references for each variant, and so on. Here is even more comprehensive report. This is actually a bit old, you know, the layout of it, it's a bit outdated. This is the latest version of the reporting part. So as you can see, you can see the mutation name, the gene, exon number, variant type, RSID, zygosity for each particular variant you decide to report. Here you can also see the information again coming from clinical data resources, information about the genes, information, you know, the list of phenotypes and diseases, mode of inheritance. It's pretty comprehensive. List of drugs associated with a given gene. And on the page 16, it's pretty long, this one. So on the page 16, you can see also the list of clinical trials for somatic samples, as well as a list of references. Yeah. So, you know, the reporting part, the report can be fully customized, you know, again, according to your needs. You just need to set up the template, and then you can use it all over again. So I think I have covered, you know, all the most important aspects. We are also running out of time, unfortunately. As you can see, it's a complex platform. You know, you start from FastQ or VCF, and at the end you get a clinical report. It's a complex platform, but once you start using it, I'm sure you will see it's quite easy to use. And as I mentioned before, VarSome Clinical is certified, you know, for clinical use. It's got a CE-IVD marking. You can process any kind of NGS data, be it a gene panel, exome, genome. It doesn't really matter. And in certain cases, you know, under certain partnerships, the VarSome Clinical comes as a part of the assay. This is what we call the bundled solution, which is the case, for example, of Swift Biosciences assays, for example, but we have also other partners, you know, participating in this program. And you can even bundle, you know, VarSome Clinical with Agilent assays through our collaboration with Perlan, our distributor in Poland. So I think I've just come to the end of my presentation. Again, thank you very much for your attention. Wojciech, thank you also very much for your help with organizing the webinar. I hope you liked it, and let's see if we have some questions here. We do. If you cannot find the essay in the drop-down list, is it possible? Yeah. So if you don't see the essay in the drop-down list here, it just means that we don't have the BET file for it. So in those cases, you need to provide us, you know, the BET file for the essay, and then we upload it to the platform, and subsequently you will see it in the drop-down menu here. What is the price without the kit? Very good question. So VarSome Clinical doesn't have any license. Yeah, with VarSome Clinical, you pay as you go on a per sample basis. In other words, you are supposed to pay for each sample analyzed. Yeah. So basically, you can use VarSome Clinical in two forms. You can use it either as the bundled solution. Yeah. So in this situation, the VarSome Clinical comes with the assay, so you pay for the assay, and you get VarSome Clinical as a part of the assay. And you can also use it as a standalone solution. Yeah. So if it's a standalone solution, we charge basically on the size of your sample. Yeah. So in principle, we charge based on number of variants in the VCF file, or in terms of the number of megabases in reads for FASTQ files. Yeah. I would, Perlan is our distributor, so Perlan is the point of contact for you to get pricing for VarSome Clinical.
Yes, thank you, Thomas. I can address that as well. Basically, please get in touch. In the case of Kraków, please contact Wojciech Bieniek. He is the person who would give you the prices, the exact prices per sample, depending whether this is the VCF or FASTQ file, and he will explain exactly what the pricing depends on. And for the other people, I'm sure that you are in touch with other members of Perlan team, so each one of them can give you all the details that you ask for.
Right. Generally speaking, that I think that our prices are quite competitive. I also actually wanted to show you, just for your information, the list of our clients, as you can see. So here you can see the list of our clinical clients in Europe. As you can see, we have a number of customers in pretty much in every European country, as well as in Middle East. And we actually have even a couple of clients on the global level. So we have quite a few clients in the US, in Mexico, in Argentina, in Singapore, Hong Kong, South Korea. So yeah, so we are actually on the market for 6 years now, and yeah, we are getting now lots of traction. So, and also another our advantage is although we have like a global presence, we like to provide personalized services. Yeah, we are always very close to our clients. Yeah, we like to support our clients on the mission as much as we can. That's also the reason why we offer these algorithmic filters, which can be fully customized as a part of the service. Yeah, so indeed, we really like to be as close as possible to our clients. Questions? Yeah, I think we have run out of questions, Wojciech, so I think we could perhaps, finish, right?
Yes, Thomas, I think that we may finish. Thank you very much for organizing this. And as Thomas mentioned, it was recorded, so we will provide you the recording afterwards so that you are able to listen to it and check it once again. And in case you have any questions, please get back to us, preferably. So because Thomas is responsible for the whole Europe, so I'm sure that he is quite busy, and we will give you all the answers that are necessary. Of course, if you wish, feel free to ask Thomas directly. He is the expert.
Yes, exactly. I don't know if I'm the expert, Perlan, they are really, they have really good understanding of the platform. And so Perlan shall be your first point of touch. However, of course, you can always, get in touch anytime you feel like it. All right, so I'm stopping the recording.